Welcome to Looping Pixels, where we explore the mysteries of the human mind. Today, we are attempting to unlock one of the most universal yet really persistently baffling secrets of our own history.
Yeah, it's a phenomenon that affects every single person listening right now. The great blank space at the very beginning of your life story.
Exactly. We call it infantile amnesia, and our mission today is a real exploration into why you cannot, as an adult, remember being a baby. I mean, have you ever stopped to wonder why you can't recall your first birthday party?
Or maybe the actual feeling of taking those first wobbly steps.
Right. That entire foundational period of your life, typically before the age of 3 or 4, it's just a total blank slate for almost all of us.
It truly is a universal puzzle, and the first assumption people often make is that babies simply don't form memories, but, well, that's fundamentally wrong.
Oh, really? So they do form memory?
Oh, absolutely. Memory formation is robust, even in the earliest months of life. The issue, it seems, is the durability of those memories, how long they stick around.
Okay, durability. Let's unpack this right away, because that sounds like the first really surprising fact in our source material. What kind of memory capacity are we actually seeing in a brand new brain?
Well, researchers use some pretty clever, specialized training methods, like teaching an infant to kick its leg to move a mobile hanging above the crib.
Ah, okay. To see if they remember the connection.
Exactly, to test retention in very young subjects. And they found that memory retention actually increases almost linearly with the age of the child at the time of training.
Linearly, so steadily increasing.
Yeah, and the absolute difference between, say, a 2-month-old and a slightly older infant is really striking. A 2-month-old infant, for example, might only retain a memory for about 24 hours.
Just one day. So if they learn that kicking trick on a Monday, by Tuesday morning, the information is already gone. That rapid decay rate is incredible.
Precisely. Now, contrast that staggering fragility with an 18-month-old infant. Even when both the 2-month-old and the 18-month-old are trained to the exact same starting criteria, so meaning they encoded the information equally well initially, the 18-month-old can demonstrate memory retention for as long as 13 weeks.
13 weeks? Compared to 24 hours. Wow.
It's a huge jump. That's almost a full season of retention.
That rapid acceleration and durability all within, 16 months? It really highlights that memory formation is anything but passive. The brain is aggressively developing its storage capacity.
Definitely. And we know definitively that toddlers can form specific event memories, the kind we rely on as adults to build our life story,
You mean episodic and autobiographical memories?
Exactly. There's a wonderful, vivid example of this from research often called the crib talk studies.
Crib talk? I think I know this.
Yeah.
Is that the charming habit toddlers have of talking to themselves, maybe to their toys, in their crib before sleep?
Yes, that's the one. A researcher named Catherine Nelson actually recorded the private crib talk of her own 2.5-year-old daughter.
Her own daughter?
Yeah.
Fascinating. What did she find?
Well, the daughter clearly described a unique, singular episode, the event of going out and buying a new doll. She even discussed the interaction, the details of the purchase.
And that's significant because, because that memory hadn't been socially rehearsed, right? Mm-hmm. Her parents hadn't drilled it into her. She encoded it herself, and she could retrieve it and describe it verbally.
Correct. The key takeaway is she had a genuine, specific autobiographical memory at 2.5 years old, a real memory of a specific event.
Yet, and here's the kicker, I assume.
Yet we know that when she grows up, that specific memory of buying the doll, along with probably 99% of her other memories from that year, will likely be completely inaccessible, gone.
That sets up our central conflict perfectly, and it's a question we explore often here at Looping Pixels. If infants and toddlers can form these specific episodic memories, why do we, as adults, completely lose access to them? Is the memory itself physically destroyed? Or is it, simply still there, just locked away behind some kind of barrier?
That is the core philosophical and biological debate in this field. And we should probably hint at the cutting-edge answer right now, shouldn't we?
Please do.
Some recent findings, particularly from neuroscientists studying the hippocampus, suggest that infantile amnesia might actually be less about destruction or a failure to encode in the first place,
Okay.
and more about a retrieval problem.
A retrieval problem. Okay, now here is where it gets really interesting, potentially a bit sci-fi. So you're saying there's a radical possibility that those early memories may actually endure in some form into adulthood, despite being completely inaccessible to our conscious minds. They're encoded,
Yeah,
but maybe the key to the vault is lost.
that's the shocking possibility, yeah. But to understand why the key might be lost, we first need to understand just how volatile even the memories we do keep actually are. We need to look at the psychological landscape of those first recollections we think we have and how much they shift over time.
Okay, let's transition then. Let's look at the actual experience of memory, the memories you claim as your own, and this idea of a shifting timeline for what we call our earliest memory. So, as we noted, the specific memories we lose before age 3 or 4 are called episodic and autobiographical memories, or AM. Maybe just quickly establish why these are so unique before we talk about how they become unstable.
Absolutely. Think of your memory system as having several buckets, let's say. One bucket holds semantic memory. That's facts and general knowledge.
Like knowing a dog barks or Paris is the capital of France.
Exactly. That stuff tends to be highly stable, generally. The AM bucket, however, holds episodic memory. Specific, personally experienced events tied to a context, a specific time, a specific place, a specific emotional state.
The what, where, when of personal experience.
Precisely. This bucket is what allows you to construct a personal, continuous narrative of you, your life story.
And it's this personal narrative that appears to have a starting point that actively moves away from us as we age. It's not a fixed date.
This is one of the most surprising findings, really. It shows that memory isn't just about storage, it's also about the conscious process of dating and retrieving things. Researchers conducted a powerful, long-term, longitudinal study.
Following the same kids over time.
Exactly. Tracking children's self-reported earliest memories over 8 years. And they discovered what we call the moving target phenomena.
A moving target. That definitely suggests the memory boundary isn't stable. How does that actually show up in the data?
Well, when children were initially interviewed, say around age 7, the average age of the very first memories they recalled was about 35.8 months, so just shy of 3 years old. Pretty early.
Okay, under 3.
But then, when those same children were interviewed 8 years later, when they were teenagers, the average age of their earliest recalled memory had shifted forward substantially.
How substantially?
It had increased to 52.5 months. That's over 4 years old.
Wait a minute, let me get this straight. If they were recalling the same event, like, say, a specific trip to the beach, they were dating it later and later in their own minds as they got older. That's kind of mind-bending.
That's exactly the post-dating detail. The children were systematically dating the event to a considerably later age as time passed. It's almost as if the adult brain, seeking coherence and stability for its narrative, unconsciously tries to push the start date of their story forward to an age when they believe they should have stable memory capacity.
Huh. That raises a huge question for us here at Looping Pixels, doesn't it?
Yeah.
If we systematically date events later to fit our adult idea of when memory should start, how confident can we be in any of our self-reported earliest recollections?
Yeah.
Does this process of post-dating sort of undermine the foundation of our entire autobiographical identity?
It certainly forces us to acknowledge that memory is a constructive process, not just like playing back a recording.
Yeah.
The brain is constantly editing the timeline, maybe to make the narrative flow more logically from its current perspective. And this systematic dating change might be partly why younger children can often recall earlier events than older children or adults can. They maybe haven't yet applied that mature filter that pushes the timeline forward.
So the boundary is moving. But you also hinted that even the content of the memories that do stick around seems to be highly unstable, not just the date.
Precisely. Even adults, they struggle with the details of their earliest memories, but they are often highly consistent in identifying the event itself. The overall structure, the headline of the memory, seems stable.
How stable is that anchor, that headline?
Very stable, actually. In one rigorous study of adult women followed over 4 years, they nominated the same event as their earliest memory, with an 82% consistency rate over 2 or 4 years. So the basic story, the time I went to Flintstone World, that part seems fixed.
Okay, 82% is pretty consistent. The anchor is fixed, but now, tell us about the details. This is where you said it gets psychologically unnerving.
Right. Here's the twist. When researchers analyzed specific content included in the narratives, the sights, sounds, specific dialogue, the minute details surrounding that fixed event.
Yeah. What did they find?
Almost 50% of the details provided were different relative to the initial report given just 4 years prior.
Half of the specific information, the textures, the dialogue, maybe the smells, is reconstructed or changed every single time we pull that memory up. That's not stability. That sounds like active, high-level editing.
Yeah.
What's driving that detailed reconstruction?
It strongly implies that retrieving an autobiographical memory is not like hitting playback on a VCR or pulling up a file. It's an active process of reconstruction. When you retrieve an old memory, your brain often seems to fill in missing blanks using your current knowledge or schemas, your current understanding of the world. You might integrate new information you've learned about that time, or maybe reshape the narrative to better align with your current self-concept, the person you are today. The event is an anchor, but the story around it changes, maybe to make more sense to the adult you.
Can you give us a concrete example from the research, something to really illustrate this dynamic reconstructive change? I think you mentioned one about a crocodile tunnel versus Fisher-Price furniture.
Certainly. This is a classic example of how unstable even the designation of earliest memory can be, let alone the details. One participant was followed over several years. In report 1, she was confident her earliest memory was being 3 years old on a family vacation.
Okay.
She recalled crawling through a long, dark, crocodile-shaped play tunnel at a theme park called Flintstone World.
Right, I can picture that.
She recalled getting extremely scared, getting stuck inside, and her brother and a foster brother trying to talk her out from both ends of the tunnel. She said it felt like an eternity.
Okay, that sounds like a vivid, emotionally charged specific event. Seems like a good candidate for an earliest memory.
Absolutely. But then, years later, when prompted again for her earliest memory, she completely abandoned that traumatic tunnel memory. Didn't mention it.
Really? What did she recall instead?
In report 2, she recalled a different memory, which she dated vaguely between 3 and 5 years old. This time, the memory was of playing with Fisher-Price little people figures in her basement.
Fisher-Price. That's a big shift from a scary tunnel.
A huge shift. She was meticulously setting up the garage structure as the bedrooms for the Partridge Family figures. Crucially, she recalled being intensely upset the next day because she had cleaned up all her toys before bed, and she couldn't remember how she had arranged the tiny plastic beds the day before to make them fit perfectly again.
So the original memory of the crocodile tunnel, which sounds like an external trauma, right?
Yeah.
Something that happened to her, was entirely replaced by a memory about being frustrated over tiny Fisher-Price furniture, an internal emotional conflict related to order and perfection.
Exactly. Her brain, years later, prioritized a memory of internal emotional frustration and a problem-solving challenge over a fixed, shared external experience like the tunnel incident. It really shows the highly subjective and reconstructive nature of our earliest autobiographical memories. They seem to be constantly rewritten, edited for detail, and even post-dated as we age.
That's a really powerful psychological framework.
Yeah.
Okay, so now that we've established that the memories we think we keep are actually unstable and kind of moving targets,
Yeah.
we need to turn to the most radical explanation for why the truly earliest memories, like the 2.5-year-old buying the doll, disappear entirely. We're moving from the psychological reasons to the core, maybe even destructive biology of the infant brain itself.
Right. We're ready to look at the brain's own mechanism for forgetting. This is where we discuss the process that sounds almost intentionally destructive, in a way.
The idea that the brain is essentially erasing its own content to make room for new growth. Is that the gist?
That the essence of the leading biological hypothesis for infantile amnesia? Yes. It suggests that IA is caused primarily by the extremely high rate of postnatal hippocampal neurogenesis.
Okay, whoa. We definitely need to slow down and define those key terms so our Looping Pixels audience can really grasp this mechanism. First, let's talk about the hippocampus. That's the star player here, right?
Absolutely. The hippocampus is this critical, curved brain structure located deep inside the temporal lobe. Its primary role is forming new long-term relational memories. Exactly the episodic kind, the story-based memories we were just discussing.
Okay, hippocampus, new memory formation. Now, neurogenesis.
Neurogenesis simply means the birth of new neurons, new brain cells.
And this is unusual in the hippocampus, or part of it, after birth.
It is quite unusual. In almost all brain regions, neuron formation, the creation of new neurons, is completed shortly after birth. But in a specific area of the hippocampus called the dentate gyrus, or DG.
Dentate gyrus. Okay.
New neurons continue to be added throughout development and incredibly even into adulthood. This is one of the very few places where the adult brain still generates brand new cells.
And in infants, that rate of new neuron production in the DG isn't just active, it's extremely high, you said.
It's explosive. Absolutely explosive. And the radical idea here, formalized by researchers like Sheena Jocelyn and Paul Franklin, among others, is that this massive, rapid production of new cells actually promotes forgetting.
Wait, promotes forgetting, not just fails to record.
Exactly. It doesn't just fail to record memories stably. The very process of creating space for these new neurons, integrating them into the existing network, seems to destabilize and ultimately clear out the pre-existing, newly formed memory traces.
Hold on. Evolutionarily, why would the mechanism that helps the brain grow and learn also be the mechanism that actively destroys the oldest information? That seems incredibly inefficient, like wasting energy recording a memory only to immediately destroy it.
That's the excellent, challenging question right at the heart of the research. The prevailing theory is that the infant dentate gyrus is functioning differently than the adult one. It's acting more like a rapid turnover system. The infant brain seems to prioritize flexibility and statistical learning over long-term stability for specific episodes.
Flexibility and learning patterns over saving specific stories.
Kinda. If you think about it, an infant is bombarded with new information every second. You need a system that can rapidly integrate new circuitry, learn patterns quickly, even if it comes at the expense of old, specific memories that might not be relevant later in a completely different context.
So the infant brain maybe chooses to be flexible and adaptive rather than stable and archival like an adult brain.
Precisely. Let's use that analogy we discussed earlier. Think of the hippocampus as a library that's undergoing massive, rapid expansion.
Okay, a library under construction.
When you, the infant, first check out a book that's encoding a memory, it gets placed on a shelf and indexed somehow in the network. But in an infant's DG, the construction crew, that high rate of neurogenesis, is constantly pouring new concrete, building new structural columns, integrating brand new wings into the library structure.
It sounds like a chaotic construction zone.
It probably is, neurally speaking. And to make room for all the new electrical wiring and structural columns, the old shelf labels, your newly formed memories, are constantly being rearranged, moved, the connections are degraded, maybe even erased, to accommodate the new neuronal structure.
So later on,
It becomes impossible later on for your adult self to find that original book because the entire filing system, the structural context that was used to retrieve it, has been completely rebuilt around the new infrastructure.
wow. So the information might technically still be there in some fragmented form, but the map, the context, the structure used to retrieve it have been completely wiped out by the ongoing building renovation. That's a far more elegant explanation than simply saying babies don't remember.
It is. And this theory is strongly supported by experiments where scientists manipulate this process in animal models.
Now, how do you do that?
Researchers can genetically or pharmacologically manipulate the rate of neurogenesis in rodents, like mice. They found that if you decrease adult hippocampal neurogenesis, if you slow down the construction crew in the mature mouse brain,
Yeah.
it actually promotes the long-lasting maintenance of memories formed after you slow it down. the structure becomes more stable, and forgetting decreases.
Okay, so slowing neurogenesis helps memory stability in adults. What about the reverse? If you artificially induce a high rate of neurogenesis in an adult mouse, if you ramp up the construction crew, does it temporarily start exhibiting infantile amnesia? Does it start forgetting faster?
That's the key prediction of the hypothesis. And yes, the results bear it out. Transiently increasing neurogenesis in an adult mouse after it has formed a memory promotes rapid forgetting of that newly acquired information.
So it temporarily transforms the mature hippocampus.
From an archivist where stable memories are formed, back into something resembling an infantile state where new memories fade much more rapidly.
That's a profound finding about the biology of memory stability. But here's something that relates directly to the kind of cutting-edge science we explore here at Looping Pixels. Can the rate of this neurogenesis, and therefore maybe the extent of our infantile amnesia, be influenced by our parents' life before we were even conceived?
You're talking about the epigenetic connection. It's a fascinating, almost futuristic link. There's research suggesting that yes, the extent of infantile amnesia might be inheritable through an epigenetic mechanism.
Epigenetic meaning. Not changes to the DNA sequence itself, but changes in how the genes are expressed, like instructions layered on top of the DNA.
Exactly. Instructions for how that DNA is read and used.
Can you break down this study that connects exercise to neurogenesis in the offspring? That sounds wild.
Sure. Researchers studied male mice who engaged in voluntary physical exercise. They let them run on wheels as much as they wanted. They were the runner parents.
Okay, fit mouse dads.
Right. And they found that these running fathers sired offspring that exhibited higher rates of hippocampal neurogenesis compared to the offspring of sedentary fathers, mice who didn't run.
Higher rates. Why would a father running more lead to more neuron growth in his offspring's brain? That seems like a huge leap.
It's complex, and the exact mechanisms are still being worked out, but it's believed to be an epigenetic transmission pathway, possibly involving changes in the RNA content of the sperm. The father's lifestyle, in this case exercise, seems to be activating specific genes related to neural development or influencing the pool of neural stem cells in the offspring in some way.
Okay, but the shocking implication, based on the neurogenesis hypothesis we just discussed, is critical here.
Exactly, since high neurogenesis accelerates forgetting.
Then the offspring of those exercising mice might actually display stronger infantile amnesia, lower rates of memory retention early in life than the offspring of the sedentary parents.
That's the logical conclusion based on the hypothesis, yes.
So a parent's commitment to fitness through this complex biological cascade could subtly influence how early or late your own first memory emerges. That's a phenomenal, mind-bending connection between environment, biology, and genetics that truly defines the kind of exploration we love doing here at Looping Pixels.
It really is. Now moving on, we have to shift the focus slightly, away from active memory destruction or erasure by neurogenesis, and towards the simple reality of physical brain maturation. Infantile amnesia isn't just about cells destroying old content, it's also likely about missing or immature infrastructure required for stable, adult-like memory storage and retrieval.
Meaning the baby brain just isn't structurally ready yet. It doesn't have the right wiring or network organization needed for decades-long storage.
Precisely. Early childhood is a critical period characterized by massive changes in functional connectivity, or FC.
Functional connectivity, meaning how different specialized brain regions communicate with one another, how they form coherent networks.
Exactly. Stable autobiographical memory, remembering a complex event, requires vast network coordination between the hippocampus, the prefrontal cortex, visual areas, emotional centers that simply isn't full developed yet in an infant or toddler. The highways aren't all built.
We've seen incredible advancements in neuroimaging, fMRI, and things like that, but I know there are massive technical difficulties involved in actually scanning infants to map this FC development, aren't there?
The challenge is enormous, yes. To minimize head movement and acquire high-quality data, infants under about 3 years old are usually scanned during natural sleep.
Okay, while they're asleep. Makes sense.
Conversely, children older than 3 are often scanned awake, maybe watching a movie or playing a very simple game to keep them still.
Right. But sleep versus wakefulness, those are completely different brain states, right?
Absolutely. In adults, we know for sure that functional connectivity patterns change dramatically between sleep and wakefulness. And the same is absolutely true for developing children. This difference in consciousness state is a huge confound.
So how do researchers possibly connect the data points between a sleeping baby's brain scan and an awake 3-year-old scan to create a reliable roadmap of functional development? How do they bridge that gap?
This is where cutting-edge computational modeling comes in. It's quite sophisticated. Researchers have to develop methods to essentially harmonize this data across different states and ages.
Harmonize. How?
They use complex computational techniques like something called elastic net regression models. In maybe simpler terms, these models allow scientists to predict what the connectivity patterns in the sleeping state would look like, based on the known, reliable patterns they see in the awake scans across different age groups.
So they're using the awake data from older kids to mathematically model and adjust the sleeping data from the younger kids to kind of level the playing field.
That's a good way to put it. They are essentially building a complex statistical bridge between the sleeping and waking brain states to try and create a reliable normative growth chart of functional brain development from birth up to age 6 or so. It's necessary to isolate the true developmental changes from differences just caused by the consciousness state during the scan.
That sounds like a huge, complex undertaking.
It is. And once they harmonize the data in this way, they start seeing clear developmental progressions in how the hippocampus and related areas process information. Specifically, they can distinguish between 2 key functional learning pathways operating within and around the hippocampus.
Okay, 2 pathways. Which pathways are those and what's the timing? When do they come online?
First, there is statistical learning. This is the brain's incredible fundamental ability to extract patterns, rules, and regularities across repeated events.
Like learning the common sequence of steps when mom puts on your shoes, or the general structure of a conversation, maybe.
Exactly. Or what all restaurants generally look like, or the typical sounds associated with bath time. This pathway, which seems to be more prominent in the anterior part of the hippocampus, develops remarkably early. It's functionally recognizable in infants as young as 3 months old.
3 months. So even a tiny baby is already recognizing patterns, predicting sequences, building these general models of the world. But as you said before, that's more like semantic knowledge, facts about the world. It's not the specific episodic memory we lose, right?
Correct. That general knowledge seems quite stable. The second pathway is for episodic memory. This deals with specific, unique, time and place tied events, remembering the specific trip to the park, not just parks in general. The full involvement of the hippocampal network, including areas like the perihinal cortex, which is needed for forming these really stable, detailed episodic memories, seems to appear much later in development, maybe around 1 year or older, and it continues maturing significantly until age 7 or 8.
So there's a logical sequential progression, maybe. The baby has to first develop a statistical, predictable framework of the world. And only then does the brain infrastructure become ready or mature enough for recording specific, unique events against that stable backdrop.
That seems to be the developmental logic, yes. Build the framework, then populate it with specific instances.
Okay, that's the neurological infrastructure story. Now let's tie this physical infrastructure development to arguably the single biggest physical developmental leap in early childhood, the transition from crawling to walking. This brings us to the embodied hypothesis of infantile amnesia, right?
Yes, exactly. This hypothesis posits that physical movement itself, locomotion, is directly tied to the fundamental development of the memory infrastructure, particularly the spatial aspects.
How so?
For the hippocampus to form stable episodic memories, especially ones tied to locations, it needs reliable sensory input to develop and tune specialized spatial mapping cells. You might have heard of them, place cells and grid cells.
Place cells and grid cells. They sound like the brain's internal GPS system. What exactly do they do?
They're absolutely essential for spatial coding and navigation. Place cells fire when an animal, or presumably a person, is in a specific physical location like you are here, signal for different spots. Grid cells, found in a nearby area called the entorhinal cortex, fire in a much more complex repeating hexagonal pattern, like a coordinate system, that helps the brain calculate distance, direction, and trajectory.
Wow, a hexagonal grid overlaying our mental map.
Effectively, yes. Together, these cells allow you to associate specific events with specific locations, and this is vital. Without stable place in grid cells, similar events, say, 2 different trips to the grocery store experienced in 2 different physical places, can start to interfere with each other, leading to rapid, sometimes catastrophic, forgetting. The spatial tag gets muddled.
Okay, and before infants can crawl, they can't tune those cells properly. Is that the idea?
Exactly. Before active self-initiated locomotion, the infant's world is largely passive, maybe constrained to the crib or someone's arms. The infant lacks the consistent, self-generated sensory input, the relationship between their own movement and the changing visual scene necessary to really tune the place in grid cells reliably.
So the system is immature.
The hippocampal system, therefore, remains functionally immature and somewhat unreliable for stable, long-term memory formation because it can't effectively differentiate or tag locations yet.
Okay, so crawling is step 1. It provides the self-generated movement needed to start making the memory system spatially reliable. But then, the infant transitions to walking. And walking involves entirely new physics, new sensory input compared to crawling. What happens in the brain then?
This is the dramatic part, according to the embodied hypothesis. The transition from crawling to walking forces a massive and quite rapid reorganization of the entire grid cell system, and likely the place cell system too. Walking changes everything for the brain's spatial mapping systems.
In what specific ways does walking change the sensory input so drastically?
Well, think about the sensory shift. When crawling, the angle between the head and the ground is relatively low and stable. The muscle movements are large and often symmetrical. The visual flow across the floor is very near field.
All right.
When you start walking, the head angle changes constantly with each step, bobbing up and down. There are entirely new muscle movements involved, especially in the core and legs for balance. And the relationship between your movement speed and the visual flow of the environment is dramatically different. You cover distance faster, you tend to look further ahead.
It's almost like switching the entire operating system of the brain's internal GPS unit. The previous calibration, learned during crawling, is now completely obsolete for this new mode of movement.
Precisely, a fundamental recalibration is required. And that transition, that necessary reorganization, is believed to be a major trigger for accelerated forgetting of memories formed before the transition.
Why? How does recalibrating the grid cells break access to old memories?
The idea is that the reorganization of the grid cells means that the complex multi-contextual keys or pointers, the specific patterns of neural activity that were required to retrieve the hard-won episodic memories formed during the crawling period, simply no longer work effectively with the newly tuned walking system.
So again, the memory isn't necessarily lost or destroyed simply because time passed, but because the brain underwent a dramatic developmental stage change in its spatial mapping system that essentially broke the access key to the old filing system.
That is the elegant prediction of the embodied hypothesis, and they've actually tested this counterintuitively in experiments.
How?
Imagine an infant learns task 1 while they are exclusively crawling. Then later they learn task 2 just as they are beginning to walk confidently. The theory predicts that task 1, the one learned with the crawling hippocampus map, should be forgotten much more rapidly once the child fully transitions to using their walking hippocampus map, even if the time delay allowed for forgetting is identical to the retention time measured for task 2.
So memory loss is tied more closely to the developmental stage change, the locomotor transition, and the necessary retuning process, rather than just the simple passage of time.
That's the core idea, yes. It adds yet another crucial layer to the puzzle.
Okay, this is fascinating. We've covered internal biology, the erasure or destabilization by new cells, We've covered physical infrastructure, the spatial reorganization due to walking and network maturation. But memory is also deeply social, isn't it? My personal history is something I share, I structure, I recount with others.
Absolutely. The final, and perhaps in some ways the most accessible piece of this complex puzzle is the social scaffolding theory.
Social scaffolding.
This theory argues that for a memory to become truly durable and retrievable over decades, especially the emotional and complex ones that form our life story, it needs external scaffolding. Specifically, it needs language and social interaction to structure the raw experience into a coherent, shareable narrative.
Okay, so autobiographical memory development goes hand in hand with language skills. You need the vocabulary to frame the event, to label it.
Significantly so. There's clear evidence for this. In preschool-aged children, researchers found a really potent correlation. They used vocabulary scores and the child's age together to predict the specificity, the richness and detail of their autobiographical memory reports.
And what did they find? How strong was the connection?
The results showed that age and vocabulary combined explained roughly 47% of the variance in how specific and detailed a child's AM was at that early stage.
Wow, nearly half. Almost half of the explanation for how detailed their memories were is tied up in just two factors: how old you are and how many words you know. It makes perfect sense, really, because you need precise words and grammar to encode, retrieve, and describe the precise context, the emotions, the sequencing of an event accurately.
Exactly, and it's not just having the words, it's how the words are used socially, particularly in conversations about the past. This brings us to the critical concept of maternal elaborative reminiscing style.
Maternal elaborative reminiscing style. That sounds important. What is it?
It essentially describes how a primary caregiver, often the mother in these studies, talks to a child about past events. This interaction is seen as a key mechanism by which memory is socialized, how kids learn how to remember and talk about their lives.
So how a parent talks to a child about the past becomes the foundation for how the child learns to structure their own past in their mind.
Yes. Mothers, or caregivers, who reminisce in highly elaborative ways tend to do three things consistently. First, they provide lots of coherent details, setting the scene. Second, they frequently label emotions explicitly. And third, critically, they help the child resolve or understand difficult emotional experiences within the memory.
Okay, so they don't just recall the event, they help interpret it emotionally.
Right. This active process is seen as essential, not just for memory socialization, but also for the child's developing emotion regulation skills. It provides the narrative structure, the beginning, middle, end, the causes, consequences, feelings, that makes the memory durable and retrievable across years.
And I guess that helps the child understand not just what happened, but why it mattered and how they felt, which makes the memory less raw, maybe less overwhelming, and more integrated into their developing self-concept.
Precisely. Let's use the specific anecdote from the research to really show the power of this structured conversation in action. Imagine a conversation between a mother and her young daughter, let's call her Rachel, about Rachel being sad because her best friend Sheila moved away to a new city.
Okay, set the scene for this elaborative conversation.
So a mother using a highly elaborative style doesn't just say, "Oh, don't be sad," or change the subject. She validates the emotion and puts it in context. She might start with something like, "Do you remember Sheila? You two were such good friends. Yes, it makes you feel sad that she moved away, doesn't it?"
Oh, yeah?
She explicitly labels the feeling, making it tangible.
Okay, validating and labeling the sadness.
Then she helps Rachel understand the cause. "Do you remember why Sheila had to move away? Remember, it was because Sheila's father had to take a new job far away." This gives a reason, an external explanation.
Right, establishing the beginning and the external cause, not Rachel's fault.
Exactly. And finally, she provides a potential resolution or a coping mechanism without dismissing the original sadness. She might say, "But is she still your friend even though she's far away?" "Yes, she is. What can we do that might make her feel happy and let her know we miss her? Should we maybe draw her a special picture or send her a happy letter in the mail?"
So that whole process takes what could be a raw, confusing, and maybe scary emotion, sadness, and loss, and structures it into a coherent, navigable memory trace. The memory becomes more durable because it now has a clear beginning, a cause, a labeled feeling, and even a potential positive action or resolution. It moves from being just an internal overwhelming state to an external, understandable, integrated narrative.
Exactly right. That narrative context helps place Rachel's emotional experience into a framework that she can understand and cope with. And research has actually shown that children whose mothers engage in these kinds of highly elaborative conversations, especially about negative emotions, are better able to manage their own emotions in challenging situations, even a year later. The structure they learn for memory retrieval also becomes a structure they can use for emotional regulation.
That connection between narrative, memory, and emotion regulation is powerful. And does the social scaffolding framework also help explain why the onset of memory, the average age of the first memory, might vary so much between different cultures? Because the values and communication styles of the culture would dictate the narrative style, right?
Absolutely. The social environment profoundly affects not only how we remember, but also what we remember and when we start remembering consistently. If we look at cross-cultural comparisons, the differences are quite significant, and they really illuminate how memory is, in part, a social construct.
Okay, lay out the comparison for us. What are the typical differences, say, between East and West?
Well, studies have shown that the earliest memories recalled by Americans tend to average around 3.5 years old. And consistent with Western cultural values that often emphasize the individual autonomy and uniqueness.
Yeah.
These earliest memories tend to be more self-focused, often more emotionally dramatic or elaborate, centering on the child's unique personal experience, feelings, and achievements or failures.
Okay, individualistic, emotional memories starting around 3.5 years. What about in other cultures?
In stark contrast, studies looking at Chinese adults, for example, find their earliest recollections typically date later, often closer to 4 years old. And the content tends to be different too. They often center much more on collective activities, family routines, social interactions, and sometimes memories focused on moral lessons or demonstrating socially appropriate behavior.
That's fascinating. So the Western culture, maybe with its emphasis on creating a unique individual identity, encourages remembering these dramatic personal firsts, while an East Asian context, perhaps focusing more on social roles, harmony, and interdependence, encourages remembering routine-based, shared, or socially instructive events.
That is the prevailing interpretation, yes. If your cultural sense of self is less defined by being a unique, separate individual and more about your role and relationship within the group, then memories might be more important for informing social behavior and imparting lessons than simply for constructing a unique personal timeline. The focus might shift subtly from how did I feel to what did we do or what was learned.
That makes a lot of sense culturally. But isn't there an even more extreme example? A culture where first memories are reported much, much earlier, really highlighting the power of social narrative.
Indeed. The New Zealand Maori culture provides a striking example. Studies have found that Maori adults report their first memories significantly earlier than almost any other group studied, averaging around 2.5 years old.
2.5. That's a full year earlier than Americans, on average. Why?
Researchers attribute this remarkable finding primarily to a very strong cultural emphasis on oral traditions and, crucially, extremely elaborative, detailed conversations about the past within the family structure, starting from a very young age. The linguistic and social scaffolding needed for forming and retrieving stable, specific memories seems to be built very early and very strongly in that cultural context. They are essentially socialized into detailed memory retrieval practically from birth.
Wow. So the way a culture talks about the past literally shapes the start date of personal history for its members.
It certainly appears to play a major role.
Okay, this has been an absolutely incredible exploration through the mind of an infant, back through our own potentially reconstructed pasts. It really demonstrates that the process of growing up is also, in many ways, a process of sophisticated forgetting, doesn't it?
It really does.
And the final answer to that initial question, why we can't remember being a baby, it isn't one simple failure point. It seems to be a perfect interconnected storm of biological, structural, and social causes all converging.
That's the key takeaway, I think. Infantile amnesia isn't one simple thing. It's this complex combination of active neurobiological erasure or instability, dramatic physical infrastructure changes in the brain's networks and spatial mapping systems, and finally, a missing or underdeveloped social and linguistic retrieval system that acts as the final lock on the vault for those earliest experiences.
Before we wrap up our discussion here at Looping Pixels, let's just quickly distill maybe the three most eye-opening revelations we uncovered in our source material today. Things that might fundamentally change how you, listening, view your own personal history.
Okay. Revelation number 1. The boundary you accept as your earliest memory is not a fixed point in time. It is, in fact, a moving target. You, as you grow older, are likely actively pushing that boundary forward in your mind. The age you assign to that first memory today is systematically later than the age you probably would have assigned to that same memory years ago. You are, in a sense, continuously editing your own origin story.
Okay, that's number 1, the moving target. What's number 2?
Number 2. Even the single autobiographical event you think is your stable, earliest memory is probably only half real, detail-wise. Even the most consistently recalled events are subject to massive reconstruction over time.
The 50% difference.
Exactly. Nearly 50% of the specific details might change every time you recall the event years later. This proves that retrieval isn't just playback, it's an active editing process, perhaps designed to integrate the past event with the person you are today.
Mind-bending. Okay, and the third revelation?
And third, the radical biological truth. A major reason your true baby memories are lost is likely because the incredibly high rate of new brain cell production in your infant hippocampus, that neurogenesis, essentially destabilized, degraded, and cleared out many of the memory traces formed during that rapid developmental stage. The very architecture of your infant brain was simply too volatile, too much under construction for long-term archival using the mechanisms your adult brain relies on.
It really makes you wonder then about the ultimate fate of those erased or degraded or maybe just inaccessible memories. We started this whole discussion by hinting at that sort of sci-fi possibility, that those memories might endure in some form, despite being inaccessible.
Right. Researchers like those at Yale and elsewhere who study the durability of early memories across childhood really do entertain the idea that infantile amnesia might be primarily, perhaps even strictly, a retrieval problem, not an encoding failure or complete destruction.
So the memories are there, but the neural map.
The map, rebuilt by neurogenesis, reorganized by locomotion, and lacking the linguistic labels, no longer has the correct address or the right key to access them.
So if that's the case, and those earliest memories of your first year, your second year, are simply inaccessible, but still enduring somewhere deep in your complex brain architecture.
It leads to a fascinating final thought, doesn't it? What new discovery, what new Neuralink technology or perhaps technique may be spurred by our growing understanding of epigenetics or grid cell manipulation or even AI analysis of brain patterns? What could one day provide the key to unlock them?
And what would you discover about yourself or about the raw experience of the world if you could suddenly remember life through the completely unfiltered eyes and developing mind of a 1-year-old?
What would that past look like?
A truly mind-bending question to leave you with. If you found this fascinating, there's so much more to discover here at Looping Pixels.
Well, we certainly invite you to join us next time as we explore another mind-blowing fact about the universe, our brains, and ourselves.